Welcome back, everyone, to Today's Professional. Today, here with Mrs. Vinita Marathe. How are you doing, Mrs. Marathe?
I'm doing good.
Thank you for taking time out of your day to come and sit with me here today. And yeah, let's get right into the questions. So, how would you say you initially got interested in your line of work?
So I do like science, and that was one of the things which I really was interested in. Just learning about new things. And out of all the subjects, whatever I'd worked so far, I did really enjoy, working with things make basically what makes everything tick, right? So science was one of the things, and then biology was the next most thing.
Mm-hmm. And how did you go more into your interest at your schooling, and how did that play a part?
Sure. So as I said, like science was something which I knew I was interested in. I wasn't sure exactly which field.
Mm-hmm.
I mean, I did start off with chemistry, and then it took I would say it was more, as I went through university and all, I got more interested in protein sciences.
Mm-hmm.
And that's what my field has been for the past 10 plus years.
Mm-hmm.
So it was more what was going out in the field of science that kind of got me interested.
Mm-hmm.
And especially with the explosion of, things in terms of what kind of diseases everybody is facing and, what kind of research is required. I think that definitely motivated me to go in this arena.
Mm-hmm. And what would you say are some interests like people in general should have when they're coming into protein sciences?
So I would say ha- just have an inquisitive nature because as in any kind of science, not just protein sciences, um, things may seem simple on paper when you are writing up your experiment, but when you're actually running one, more often than not, it's a failure. But you have to use each failure as, what's the next question I can ask?
Mm-hmm.
What did go wrong? So I think that kind of attitude is definitely needed in any kind of science, I would say, any kind of research. It's more that you don't accept the failure, you kind of work around it and try to do better next time. I think that's the most important lesson.
Mm-hmm. And how would you say your master's in analytical chemistry and your master's in biochemistry have helped you in your workplace?
So since my focus was analytical chemistry, I do have an expertise in instrumentation, right? So sometimes people just have one aspect of it. So they would do fundamental cell-based research, if I just want to give an example. But
Mm-hmm.
since I have a chemistry background, that helps me understand one avenue. And then since I moved into protein chemistry, that helps me really, uh, I would say, jump that bridge. So when I'm working at a problem, I can use I would say both my work streams, whatever I've learned. It's not just one from a different perspective.
Mm-hmm.
So that's important. I mean, because biochemistry as such may not delve into instrumentation.
Right, right. And how would you say throughout your career, and when you started at San Francisco State, you were involved in conducting research studies, and you did a study on the development of methodology to identify cell surface glycoproteins. Could you walk us through what you did in that study and why you did choose that study?
Sure. I mean, that's actually one of the most interesting things I would say I've done.
Mm-hmm.
The first time I had working in protein chemistry. So similar to what I mentioned before, my mentor had predominantly worked in biochemistry, so molecular biology at that point. And when I did start working with him, I did mention utilize instrumentation, but apply it to the field of biochemistry. And so um, it was a really cool project where I had to start something new in the lab. This was a novel project for my mentor as well. So the idea was to look at glycoproteins, which are usually on the cell surface, or they're called plasma proteins, and very difficult to, I would say, isolate from the cell.
Mm-hmm.
The idea was that how can we use that entire plethora of proteins on a cell to, kind of defeat cancer. So the idea was to use HeLa cells, which are cervical cancer cells, and come up with a methodology where we could actually collect the plasma proteins,
Mm-hmm.
test it through mass spectrometry, and then finally apply. And this data could then be used by further, like additional researchers to look at target proteins. So the idea was to see, can we come up with something simple where
Mm-hmm.
everybody else can use? And so that was a really cool project in
Mm-hmm.
that sense.
And in this project, what would you say are some challenges you faced along with you're saying the separating of the cells which you needed to have? What is your
Mhm.
some other challenges alongside that you had faced, and how did you overcome them?
So some of the, I would say I will give you a simplest example where, so each of that method development was, I would say, 5 to 6 different steps.
Oh.
So it wasn't just, how do you pulverize a cell, right?
Mm-hmm.
But once you do that, how do you separate what you want from
Mm-hmm.
all the junk which is in there, right? And so one of the aspects which I had to work on was once I wanted to purify my product, uh, you know, what was the mechanism? So there are different solvents. I mean, I'm going into details, but if you have to get a solvent to, have a column chromatography, it's a way to separate proteins
Mm-hmm.
of interest, and it needed to figure out what was the best solution to elute the proteins of interest, because it's not one size fits all. You have to figure
Mm-hmm.
out different chemicals, different solvents, and at the end of the day, which solvent will work best to
Mm-hmm.
get the protein of interest. And I would say, in simplest terms, that was the most difficult part,
Mm-hmm.
because if you use the wrong solution, you end up with a really debris material, right? You have a
Right,
folded
right.
protein. I would say improperly folded protein. So it's kind of a waste of an experiment. So that was one of the challenging aspects.
Mm-hmm. And after you were able to get through these parts, how would you say the results were differed from what you expected at the beginning of the study?
So at the start of the study, I was pretty naive. I thought, yeah, I would, it's so easy. We'll just break the cell apart, run it over a column, and get it done.
Mm-hmm.
But it really helped me understand, the challenges, right? I mean, in a broader perspective, when we think about, cancer therapy, right? Cancer has been around for a long time, but
Right.
we're still not there. It's just that the nuances, right? The simplest thing, you really have to work hard. Maybe at the end of the 100th experiment, you realize, oh, it was that simple, but it
Mm-hmm.
takes that long,
Right,
and
right.
as I said, coming back to that, you have to take each failure of your experiment to write a better one next time. And that really helped me. That's also helping me now in
Mm-hmm.
my work, where, as I said, not every experiment works. So you just need to
Mm-hmm.
go back and forth every time.
Right, right. And what would you say are some studies you are currently doing right now?
Um, Okay, so not going in, obviously, some of the stuff is proprietary, but
Right.
so
Mm-hmm.
I am working on vaccines, right? I'm working on not prophylactic, so like flu vaccines. I'm working more on therapeutic vaccines. So we work on a vaccine approach to, I would say, resolving cancer, different kinds of cancer
Mm-hmm.
applicable to that. And so one of my the projects which I'm working on now is to uh, so once we dose patients, right, we may dose them with different combinations. So we need to know in terms of, what's the impact? What is there anything residual of the drug
Mm-hmm.
present in, the patient's body? Because you need to know, what's the effect, impact, side effects. And
Right,
so
right.
right now I'm trying to develop a method where we can take, a sample from a patient
Mm-hmm.
and be able to test even minute amounts of the drug in the bloodstream. And
Oh.
so that does require, as I said, it's minute amounts. It's not a huge amount where it would be easy to track it, right? So kind of the idea is that can we really develop a sensitive method
Mm-hmm.
to track less than 0.01% of the drug.
Mm-hmm.
Um,
And in the past and through all of your studies, what would you say is the what would you say what is your most favorite study you've done? Like the one where you enjoyed the most?
um, I have, there've been a lot, but I would say at work, um, it's been just that. I think one of the methods, right when I joined Pfizer, I think one of the things was I had to take a method which had been used in the department for the past 5 or 6 years,
Mm-hmm.
and I kind of take that as a challenge and turn it around,
Mm-hmm.
to improve it further. And so I think for me, I had the most fun with that, where I knew some things were already existing, but to really research what developments had been done
Mm-hmm.
in science and apply it to that. I think that was one of the most challenging and most fun. So it was, again, as you can hear, my background is development of methods
Right,
and assays, and that's really what I enjoy. And it comes back to the fact that because I have both chemistry and biochemistry, it helps me, kind of bridge that gap
right.
every time I'm looking at a problem.
Mm-hmm. And along with these skills, technical skills you have from your degrees in biochemistry and analytical chemistry, what would you say are some non-technical skills which are needed to be successful in your line of work?
Um, I think as you grow high into the industry, once you're starting, like fresh in the industry, yes, obviously it's your
Mm-hmm.
educational background. But as you go further, I think it's important to I think just be sincere about your work in that sense. But then as you go more into leadership positions or managerial positions,
Mm-hmm.
it's good to have good personal skills, like interpersonal skills.
Mm-hmm.
Be able to not just do your work, but be able to lead others.
Mm-hmm.
How to manage, how to help them do their own work without micromanaging, because that's
Mm-hmm.
important.
Mm-hmm.
Because you can't do everything, right? So you can't just write your own experiment. And so I would say the ability to let people make their mistakes and learn from their mistakes is important, rather than um, be a helicopter manager, if you would say, trying to get into it. So I think that is important as you go through those
Mm-hmm.
kind of, I think it's important to develop leadership skills. That's important.
Mm-hmm. And if you can, could you please explain your day-to-day responsibilities which you have as a principal scientist at Pfizer?
So a lot of my work nowadays is kind of behind the desk.
Mm-hmm.
So as I said, I have a group of associates who work for me, and at the I would say during the day, not every day. The plan is that if they are running their own experiments,
Mm-hmm.
I make sure, without micromanaging too much, that they are thinking about the experiment correctly. So kind of look over their plans, try to understand what different kinds of experiments they're planning, guide them,
Mm-hmm.
that maybe that's a better way to try something out, and then also work on my own experiments at the same
Mm-hmm.
time.
Yeah.
And then at the same then unfortunately there are too many meetings with other managers and all. But throughout the day, I think it's pretty much a split between experiments and I
Mm-hmm.
would say a little bit of more bureaucratic work.
And how would you say your work-life balance would be because you're handling the duties of a principal scientist? Does that carry into your home life? Like, do you have to bring work home and be able to do it there?
Not usually. So Pfizer is a good employer in that sense that we do have, and we do stress on work-life balance. We actually
Mm-hmm.
have a Pfizer work culture, which I was also a part of, where we consciously try to maintain that. Unless
Mm-hmm.
it may be a one-off chance where it's a deadline and you really have to get things done. But otherwise, the expectation from everybody is that once you go home, you
Mm-hmm.
just close everything down. I mean, it doesn't really happen, but it's not the expectation.
Right.
That's the most important thing. That the expectation is that you go home, relax, take your time, and come back recharged.
Mm-hmm.
So I think overall, they have a very good work culture in that sense.
Mm-hmm. It's always important to have like a good work-life balance so you can
Yeah,
be your best at work and be your best at home.
exactly.
And I noticed that before working at Pfizer, you worked at San Francisco State and Novartis as well. So how would you say
Hmm.
your experiences at these companies have helped you?
So San Francisco State was really fun. The idea was, I mean, of course, that I was working in my mentor's lab
Mm-hmm.
again, and so that was more of a university setting. And I would say at that point, I really wasn't thinking of myself as
Hmm.
Yeah, like an employee as such, but it was fun. So it was much more relaxed. Obviously, as I said, it's a academic institution
Right.
rather than an industry. And then at Novartis as well, this was also significant. So it was pretty good. So my experience in Novartis was actually even in Switzerland. So I
Oh.
wasn't, so then there again, the work culture is different,
Hmm.
but I think Europe definitely has a better work-life balance than the US. So I would say it was even better.
Hmm. And another thing which I noticed is that throughout these two companies, you went from scientist one to scientist two to an analytical senior scientist to principal scientist. So how would you say
Hmm.
your responsibilities differed between these positions, and how
So yeah,
and
yeah, go ahead.
yeah, and how have those experiences helped you?
So when I first joined Novartis, again, this was after my second master's at San Francisco State, and so
Hmm.
I wouldn't really say I was a completely junior level scientist, but midway through, right?
Okay,
So at
right,
that
right.
point, I was still fresh to the industry,
Mm-hmm.
and so it was more of, I would say, definitely more lab work. I had my own manager, who then do the things which I'm doing right now, right?
Mm-hmm.
He would support me in my experiments. I would plan my experiments, talk to him, and kind of go from there. And so I looked at him for guidance on the inferences of my experiments and
Mm-hmm.
kind of to help me plan the next ones. Although you had to come up with your own ideas, and so a scientist who again was a progression on that, where you tend the idea is that as you go further up, you become more and more independent. So you should be able to not just plan your experiment, but already have some idea what the conclusion would be,
Right,
right?
right.
I mean, it could end up not working, but at least you should have some idea of why are you doing that experiment. And so each of these steps to where I am right now, the underlying principle doesn't change, right? You still
Mm-hmm.
are involved in science, experiments, etc. But you're at the stage where now, even without physically running an experiment,
Mm-hmm.
even on paper, you can visualize this is what will happen. This will, and those are the instances. So in some cases, as I moved on to a principal scientist position, I know it was more the industrial experience rather than just the educational background,
Mm-hmm.
where I'm at a point where, I don't really need to run an experiment, say, to get an idea, because
Right.
that knowledge through the years has helped me. So I think the progression is more that you go kind of more and more into, you become more knowledgeable. That's what I would say.
Mm-hmm.
So it is not just the position name changes. It's
Mm-hmm.
like being in, if you're in, simplest example is if you start off in kindergarten
Mm-hmm.
and you're in high school, right? So that's
Yeah.
your progression right there.
Right.
So if things which were difficult in kindergarten won't be as difficult when you're in high school.
Mm-hmm.
So that's the best way to describe it.
Mm-hmm. And how would you say your field has changed from when you initially got into it versus how it is right now?
Um, Not even, yeah. So I kind of moved around. Although it's protein sciences, I started off working with monoclonal antibodies, and so those were one of the big things. Again, I was always working either in some kind of cancer therapy or, um, you know, uh, more so like specifically monoclonals, and those are different from a vaccine industry. So
Oh.
vaccine is
Mm-hmm.
much more complicated than running just doing cancer therapies in that sense. So when I first joined Pfizer, it was a learning curve for me. Not so there are surprisingly to hear vaccines are so complicated because there are a lot of things
Mm-hmm.
which go in the background,
Right.
and that I would say for me has been the biggest change. So even in science, I have jumped from one kind of stream to the other.
Mm-hmm.
And again, it's not the same. It's like you have the same similar principles, but it's a different technology. It's a different experience, I would say.
Mm-hmm. And as you were saying that vaccines are really difficult to make and understand. So if you could give like a simple rundown of how a vaccine is normally made.
So you start off with obviously you have to grow something in cells, right? So
Yeah.
you have to figure out which kind of cells.
Mm-hmm.
You have different kinds. You usually a human cell line, and then you, depending on what kind of vaccine you're looking for,
Mm-hmm.
right? I mean, the simplest example now is the Pfizer vaccine for COVID and the Johnson and Johnson vaccine, right? So they have different technologies.
Mm-hmm.
So the Pfizer one, I mean, that's the best way to kind of explain it to you. So you need to figure out what is your um, a control vehicle. What kind of virus do you want to have,
Right.
right? What kind of technology? And so you actually infect. That's the best way to describe. You infect the cell line with
Mm-hmm.
the virus of interest.
Mm-hmm.
Then you let it. Usually, it requires a couple of days to grow
Mm-hmm.
or infect or proliferate, and
Right.
then you uh, I would say simplest is you purify the virus from the cells,
Mm-hmm.
and that's where technology comes in place, where you have different mediums, chromatography, etc.
Mm-hmm.
So you purify that, and then it's to purify. Need to know what solution it needs to be in, right? I mean, that is
Mm-hmm.
important. Otherwise, you'll end up
With something.
getting a yeah, it's a glob, right? And
Right,
if you
right.
don't have the correct solution, it will just like precipitate out.
Mm-hmm.
So you need to know what kind of solution do you want, and that's your crude product, right? Then you need to purify to get rid of all the impurities from the cells, which are also coming along the virus,
Mm-hmm.
because at the end of the day, you want a clean product, right? As clean a product as possible.
Mm-hmm.
And so you purify it along the way, and then so this is what a process group will do, a process scientist will do,
Mm-hmm.
who do cells technology. And then from that end, my group will support them along the way by running assays to figure out, what is the concentration of the virus, what are the impurities, how many are there, is it enough, does it pass regulatory guidelines? If
Mm-hmm.
not, you go back to the drawing board and you make changes. So that's like the entire simplest term. That's kind of the process by which you make a vaccine product, right?
Mm-hmm.
And so in terms of solution, it is important because not like one, like water, and it's like a buffer which
Right.
may have 5 6 different components, chemicals in it,
Mm-hmm.
like a surfactant, etc. So you need to know what is the percentage of surfactant,
Mm-hmm.
what concentration, and so it's every detail goes in place to get a good final product at the end.
Mm-hmm.
And so your purification, so it's like you start off with the cells and you end up with a purified product. So
Mm-hmm.
that's the I would say the best way to describe the simple process that we make.
Yeah. And you were talking about how your team and you have to run tests to see if it's within regulatory guidelines. What is the need of these regulatory guidelines and what are those guidelines?
So it's important, right? Because at the end of the day, you are going to give this medicine to people.
Mm-hmm.
Prior to that, you're even going to test in animals, right? You have to do
Right,
this. So you
right.
can't test in humans directly. And so it is also ethics, right? At that point, you can't
Mm-hmm.
just inject something.
Mm-hmm.
And this has come through the guidelines through a long time, right? So for certain kind of impurities which we know are going to be there in your final product,
Mm-hmm.
it is, hold on one second. Yeah,
Yeah.
it is going to be, hold on, my dog is kind of going crazy now.
Yeah.
So that idea is that um, because you want your product to be within a certain guidelines, right? So
Mm-hmm.
you know that if your final product contains X percentage of these ingredients, it is fine. It is safe for
Human
injecting
use.
into a human being,
Mm-hmm.
right? So that's the regulatory perspective, that you need to be as safe as possible.
Mm-hmm.
And that's important, right? Because you don't want to inject something which is going to have more side effects or is going to be harmful than actually being useful.
Right, right.
And those guidelines, as I said, through the years, as the FDA has looked at different
Mm-hmm.
products, different processes, that's how they've come up with these guidelines. And it's more of um, through the years, you kind of track the products and you see what is acceptable.
Mm-hmm.
If it's something new, then obviously it takes a few more years, right, to figure out
Right, right.
what is the most acceptable limit. So till then, if there's a new process, then the FDA usually gives the most strictest guidelines,
Mm-hmm.
so because you don't know, right, much about a process. So it
Mm-hmm.
starts off with really stringent guidelines, and then more and more data, then you get instead of 1 to 10, you can go from 1 to 100,
Mm-hmm.
right? So that's what, and that's the importance. Like, it's not just making those products, it is important that they have to pass a certain criteria.
Mm-hmm.
So that's where my group also comes in, where it's the process group will actually generate the material, but
Oh.
you need to know that at the end of the day, that is it worthwhile,
Mm-hmm.
taking it forward.
Right, right.
Because that's one of the things, right? I mean, the reason why it takes about almost 8 years for something coming from a lab to a market is
8 years.
that there are usually 100, yeah, more than, I mean, 8 years is a minimum. And that too, you may start off with 100 molecules,
Oh.
but only one of them will end up going to the market because most of them will fail at some point.
8 years is a long time.
Yeah, I know. And this is the reason why we still don't have a therapy for cancer, right? Because it may be the best in the lab, but if you can't make it in a form where you can actually inject it in humans, then it's useless,
Right,
right? So which
right.
is why it's
Mm-hmm.
not that it's not there.
It's just that we have to make it so that people can actually use it.
Yeah, exactly. Yeah.
Yeah. And as you was talking about the COVID-19 vaccine, COVID-19 has been a big part of the world over the last year or so. So how would you say
Mhm.
it has affected your line of work, other than like obviously Pfizer's been making the vaccine, but other
Yeah.
than that, how has it affected your line of work?
So I think we've been okay. I mean, it does impact. It's funny because we've been talking even in our labs that even if you don't work on a COVID-19 project,
Mm-hmm.
it does impact how we perform our, day-to-day experiments because it's in terms of work, it's not impacting us too drastically because we have the flexibility of working from home.
Mm-hmm.
So our work hours, obviously, Pfizer has been really good where we maintain social distancing, which
Mm-hmm.
is difficult in a lab, but still have to work. It's
Right.
difficult wearing masks and everything and,
Doing the
getting all that
experiments.
stuff done. Yeah. But then in terms of, I would say, because everything is prioritized for
Mm-hmm.
COVID-19 in terms of, materials, right? What we need for experiments. So
Mm-hmm.
it gets difficult to even get stuff from the market to get our own experiments. So that is a downside, I would say.
Mm-hmm.
So you have to be really creative,
Mm-hmm.
but I think everybody's in the same boat. You have to be flexible about how you're working in this
Right.
environment.
Yeah, everyone got to be safe so that this doesn't last another year or
Yeah,
two. And then you were
yeah.
saying how your team works on the regulatory guidelines to make sure everything is intact, to make sure it's safe. What do you say were some challenges other than meeting these guidelines in making the vaccine?
Um, I think the regulatory aspect does play an impact, but I think I'm trying to figure out one which would be easier. But if you're starting with something new, sometimes it's easier, sometimes it's very difficult. But
Mm-hmm.
you, I think the part where I would say when you're purifying a virus, right,
Mm-hmm.
for the vaccine, you need to know, as I said, what is it most stable in, right? And sometimes that takes, you may have guidelines from, papers in literature where what people have used before,
Mm-hmm.
but you do not know till you actually test it out whether it will work in your process,
Right,
right?
right.
And so there are different things where you may have to tweak a filtration somewhere,
Mm-hmm.
right? You need to
Right,
filter
right.
it in a certain way. You need to store it in a certain way.
Mm-hmm.
So one of the things what you see here is the Pfizer and Moderna vaccine are stored at -80, and the Johnson and Johnson at 2 to 8,
Yeah.
right? So that again, that tells us
Mm-hmm.
as scientists that okay, they're formulated in a different way, which is why
Mm-hmm.
they can be stored at two different conditions.
Right, right.
There is a reason why those two vaccines cannot be stored at 2 to 8. What it means is that they are not stable, right? And so these are the things which um, that is the science behind. It's not just people picked up
Mm.
-80 and 2 to 8. There's a reason behind
Right,
why
right.
it should be in that. So I think those are the challenges. I would say, trying to figure out what is the best, um, I would say, storage for a vaccine as well. That comes at the later stage. You have to test it out. You have to test out, and we do these studies.
Mm-hmm.
So typically we call this as formulation stability study. So
Mm-hmm.
once we actually prepare the product, we put it on different conditions for
Mm-hmm.
stability, and we track it over sometimes over a period of 2 years.
All
to
right.
see how stable it is. Yeah, so that's right.
Oh, 2 years. Okay, that's long.
So, I mean, you do make a decision after 3 months, but you still track it. So then, if 6 months you see something going wrong, you have to go back
Mm-hmm.
and change the formulation. But it's more than that. If you put enough guidelines in place, you know that you can use the 3-month data and extrapolate and say, yes, it will be stable for 2 years, but you still have to track it.
Mm-hmm.
And
Right.
that's your data which you present to the authorities when you apply for approval, right? That yes, a product is stable at room
Right, right.
temperature.
That's true. That's true. And then you were talking about how, again, the Pfizer vaccine, you have to store it at -80, and the Johnson Johnson is at 228. So, again, these are not temperatures which you normally find on Earth unless it's like in a controlled environment. So, what happens if the vaccine is not in those temperatures? Does it just go to waste? And what happens to those vaccinations?
So yes, that's as I said before. There's a reason why you have to store, at a particular temperature, because in the background the researchers have tested
Mm-hmm.
at what temperature your vaccine is going to be stable,
Right.
right? Because what happens is it may be you may have a range of, it's not like the minute you take out something out of -80, it's going to go bad,
Right. Right.
right? There's usually a time frame where you say, okay, maybe for one day at 2 to 8 it will be fine,
Mm-hmm.
but it has to be used within that time. And the perspective behind that is data. There is data to back up that after, I would say after almost one day, you would start seeing decrease in the efficacy of the vaccine, right? So
Mm-hmm.
if the vaccine, and this is just a general example, right?
Mm-hmm.
Nothing specific, but the idea is that, say, your data has shown that at say, day one, you have 90% efficacy, then but storage at 2 to 8 after one day or 2 days drops it down to 50%, right?
Mm-hmm.
And what happens is when you say drop in efficacy means the virus is kind of simplistic terms is going bad, right?
Mm-hmm. Right.
So
Right.
it's not as effective,
Mm-hmm.
right? And then there are other things where it's not just it's dropping out of the solution, it's like precipitating out.
Right.
But now if it's maybe injected in the patient, it would cause more harm than usefulness.
Right. Right.
So that's the reason why you have to specify the temperatures. Usually, as I said, there's a range,
Right. Right.
right? But then
Mm-hmm.
that's the reason why you have to keep it at those temperatures. And if you still have it at a wrong temperature post those days, you have to discard it
Mm-hmm.
because you no longer can safely use that
On humans.
vaccine.
Right. Right. And then, does your company Pfizer have any internship, volunteering, research opportunities for high school, college students? If so, could you name some of them for me?
So there, we do actually have a summer internship program. I
Mm-hmm.
think that's literally what it's called. If you go to the Pfizer website, it's called summer internship, and
Mm-hmm.
it's mostly for, if I'm not mistaken, post junior year high schoolers.
Mm-hmm.
To college students, we do but that is more on an individual lab basis. So we
Mm-hmm.
have student interns who are start off in their sophomore year in university
Mm-hmm.
and work in our labs for the next 3 years till they graduate out of
College.
the year. Yeah,
Mm-hmm.
but then that's a different. That's like a paid internship.
Mm-hmm.
Like you're
Right.
instead of working somewhere else, you're working in the labs with us.
Right. Right.
The summer internship is just for that's what it means, just during the summer for high schoolers,
Mm-hmm.
to see if this is something they're interested in pursuing.
Mm-hmm. Yeah. And then, as we were talking about before, some interests which you should have to be in this field. What would you recommend high school students should take? Like, what courses should they take? What books should they read to become more knowledge, to gain more knowledge in this field?
Um, I, I'm just trying to figure out, but yeah, I would say basic understanding of biology.
Mm-hmm.
Uh, chemistry is important. I would say chemistry and biology both, because um, but then I think the newest field nowadays, and I think most of, I'm not sure if this is taught in high school,
Mm-hmm.
but immunology is like the main underlying concept
Right.
of most of these, the direction in which these pharmaceutical companies are going, especially biologicals, right? So
Mm-hmm.
when you say pharmaceutical, you also have small molecules like Tylenol, those over the
Yeah.
counter. So that's still
Mhm.
pharmaceutical, but it's a different field. It's called small molecules.
Mhm.
And when you work with proteins, it is called biologics or large molecules.
Mhm.
Right. So for those, you do want to read things on fundamentals of immunology, because I think that's where I feel the field is going.
Mhm.
And if you're if that strikes sparks your interest,
Mhm.
that's a really cool thing to do. I know most of you kids in high school are interested in the CRISPR because it did get the Nobel Prize
Yeah.
last year.
Mhm.
So that was again a cool technology. So
Yeah.
I would say immunology, if nothing else.
Mhm. And yeah, those are all the questions I have for you today. Thank you so much for sitting down with me and taking time out of your day. And if you have more questions, I'll leave my email down below. Just shoot me an email and I can answer to my best of my ability. And yeah, bye.
All right, thank you.